photosynthesis. How do we improve photosynthesis? I mean, that would be very awesome if you think about it. On one side, plants uptake carbon dioxide, which we hate for the environment, and they transform it into oxygen that we breathe and into food that we need even more with the population growing. But how do we improve it? There are actually several limitations in the photosynthetic reaction processes that we can work on. And one of the main ones is actually the difficult diffusion of carbon dioxide through the plant membranes until it can reach the photosynthetic reaction centers. And this is where the field of plant nanobionics can actually help. By combining the plant natural functions with the amazing properties of nanomaterials, the field aims to enhance said natural functions or give the plants completely new ones. In our case, we focused on developing nanoparticles to solve the CO2 diffusion problem. We developed specific nanoparticles based on PEI, which is an organic polymer that is able to capture CO2 from the atmosphere and can also cross plant cellular barriers. And we functionalized it with chitosan, which is a natural polymer that is biodegradable. We functionalized it with chitosan in order to render our nanoparticles phytocompatible. We also made them green fluorescent just so that we can see them. And so at first, in test tube tests, enzymatic assays, we showed that not only the nanoparticles can uptake CO2 efficiently, but they can also increase the conversion of CO2 to sugars by 20% in comparison to just water. And then we demonstrated that the nanoparticles not only can be infiltrated inside the plants safely without damaging the leaves, but they also localized everywhere inside the cells, and more specifically at the very location of the photosynthetic reaction centers, placing them right perfectly for a CO2 concentrating mechanism inside the plant's cells. However, working on full plant models can be quite tricky. And the current infiltration methods are not really adapted because they are really localized. So it's very difficult to quantify the actual effect of the nanoparticles on the photosynthetic efficiency of the plants. This is why now we reduced the scale of our models to plant cells in suspension cultures. And in our most recent results, we actually demonstrated that the nanoparticles can reach the photosynthetic reaction centers and do not damage the cells, but they also seem to have a special affinity for the cells and modulate their CO2 exchange dynamics and growth in the cultures. So this is a very promising approach to increase photosynthetic efficiency, but we need to confirm the results, but we can already imagine the applicability of such cells to develop smart, responsive materials. And work toward a greener future where we can capture CO2 directly from the atmosphere and store it. Thank you very much.